Assalamu alaikum, everyone. I hope all of you are doing good and healthy. A very warm welcome to all of you. Thank you so very much, Pharmacozyme, for having me here and giving me such an incredible opportunity to host this event. We have Sir Farid, Dr. Farid Hasan, who is currently working as chairperson of Department of Pharmaceutics in the University of Karachi in the Faculty of Pharmacy and Pharmaceutical Sciences. Sir will enlighten us about the quality by design, which is very critical and vital importance in pharmaceutical development. Sir, please give us your introduction and over to you, sir.
Assalamu alaikum, dear participants. First of all, I appreciate Pharmacozyme and their team for organizing this important webinar today and giving me an opportunity to share some of the information related to quality by design. I welcome all the participants from Pakistan who are attending this webinar and also those who are attending this webinar from abroad. It is worthwhile to mention here that beside Pakistan, many participants from other countries that include USA, UK, Canada, Japan, Bosnia, Philippines, Bahrain, and Kenya, and other countries, they are also attending today's session. So this indicates the curiosity and this indicate the interest of the participants, not only from Pakistan but also from the other countries. So I welcome all of you and appreciate the interest. And we all, as the pharmacists, we need this interest to enlighten our profession. I hope that you will learn many things from today's webinar, and this will help you in advancing in your professional career. Regarding myself, as Abdul Hasib already told you that I am serving as an associate professor and chairman at the Department of Pharmaceutics, Faculty of Pharmacy and Pharmaceutical Sciences, and also got an opportunity to complete my education from Pakistan, perform MPhil and PhD from Karachi University. And after that, I started my career from the pharmaceutical industry and served in Gas Pharma and later in the Roche Pharma as well. And then I joined academia and I also got an opportunity to serve in various well-renowned universities such as Bakai University, Ziauddin University, Suhail University. And in 2006, I joined Karachi University as a lecturer in the Department of Pharmaceutics. I also got an opportunity to serve in the Al Marfa College of Science and Technology, which is now Al Marfa University in Saudi Arabia. So this is a brief introduction regarding myself, and I got an opportunity to work in teaching and administration, in research, and now my focus is mainly to share my information and my knowledge as far as possible. And again, appreciate the efforts of Pharmacozyme and other platforms who are working from Pakistan and from other countries as well in disseminating the knowledge and the experiences of the experts. Thank you so much. Now coming towards the webinar slides, and in the name of Allah, the most gracious and most merciful. So this is the topic of today's. So this is our topic: quality by design, a pattern from formulation and product development, and we will try to learn main key concept of this particular topic. So these are the contents of for today's webinar. Why quality by design? So this is the most important question, that why we are moving and why we are switching and why we are focusing and emphasizing on quality by design. So we'll discuss something related to this particular topic. Then we'll discuss about the elements of the quality by design, such as quality target product profile, abbreviated as QTPP, critical quality attributes, CQAs. And when we talk about the critical quality attributes, we will learn something related to the formulation of the oral dosage form. So for this, it is further divided into three subheading: drug product, material, and process parameters. Then we will discuss something about the risk assessment, Ishikawa diagram, and how we assess the risks in terms of the FMEA, that is the failure mode effect analysis. Then we talk about something about the design space, and I will try to give you the concept of the design space in terms of the design of experiments. Finally, the control strategy and the continual improvement. And last but not the least, we will discuss something about the overall session and the learning which we did. Now, why quality by design? So this is the first slide, and let us now starting from what is quality. So the first one definition of the quality, it is the totality of feature and the characteristics of the product. And we know that the pharmaceutical industry is struggling from day one to provide a quality pharmaceutical product. But at the same time, the industry is also facing various sources of variability that include the API variability, excipients variability, process and packaging variability. So all these sources of variability that are properly encountered by the pharmaceutical industry, this lead to a poor quality of the product until and unless it is not properly controlled and monitored. In the recent years, we have seen that the pharmaceutical industry as well as regulatory authorities are focusing more on the quality as compared to the previous decade. And this can be visualized from the various regulatory guidelines such as the ICHQ8 pharmaceutical development, ICHQ9 quality risk management, and ICHQ10 pharmaceutical quality system. And the purpose of all these guidelines is to harmonize the implementation of the quality practices in the pharmaceutical product development and to ensure us with this to use the systematic tool as we are entering in a challenging 21st century quality initiatives. Now, if I talk about all these guidelines, I will focus mainly on the Q8R document, which is Q8 document, sorry, Q8 document, which is the parent document, and it has provided a systemic framework for the development of the pharmaceutical product. And we all know that the second revised document which we got from the regulatory authority is the Q8R2. The purpose of this Q8R2 document is to clarify the concepts of the concepts of the parent document. Now, the principal aim of the pharmaceutical scientist is to develop a quality product that meet the pharmaceutical industry requirement as well as the patient requirement in terms of the quality, safety, or quality efficacy and safety. And one phase we come across several times that quality should not cannot be tested into the product. This is the very important top point. The quality cannot be tested into the product. That is, the quality should be built in design. So by this phrase, we mean that quality is not something which we get at the end of the formulation. Once we get the formulation, once we fabricate the drug, and now if you want to check the quality, this is not the time to check the quality. The quality should be integrated from the start of the development process in terms of the formulation, in terms of the APIs and the excipient, in terms of the processes. So later and during the manufacturing, we inspect the product, we do post production testing. So we know. So by this we mean that we have to integrate the quality into the design of the pharmaceutical product and simply check it that whether we are moving in the same direction or the right direction by testing from the start, in the middle, and at the end of the pharmaceutical development. Similarly, another important thing is that, and if you have read all, that the regulatory authority do not bound the manufacturer to use an empirical approach or the systemic approach. So this may be due to the availability of the resources and the experts. But if we compare the conventional approach and the quality by design approach, for example, if I talk about the conventional approach and OFAT, which is one factor at a time approach, you will find that in that approach, we are fixing the quantity of one variable. We are actually varying the quantity of one variable and fixing all others. So this will not give us a detailed information. This will not give us that whether there is interaction or not, or whether or whether it is suitable or not, because it varies at the quality of the excipient and the API vary. Comparatively to this, if you compare it this with the quality by design, we find that it is a systematic approach. It is based on the design space. It is based on the on the risk assessment. It is based on the on understanding the quality attributes of the product and the process and the parameters. So this is a systematic approach which is based on the statistical designing, and therefore we are more confident. So we all know that once we get a design, and once we get the concentration, that is once we get the predicted response, we will actually do the same experiment and then check it that what is the predicted response and what is the actual response. And in this way, in most of the cases, we get the same response as it is predicted by the statistical design. So this is very important, and I think that you understand that this is quality by design is a very important approach, and by this we are going to integrate the quality into our product, which is very important. Now coming towards the next thing, that what is actually the quality by design. So this is the definition we got from the regulatory authorities. Quality by design is a systematic and scientific method to the pharmaceutical development, concentrate on the product and the process parameters. So we are concentrating on the product and the parameters that impact the quality. It is based on the predefined objective risk assessment. As I already told you, it has to get improve quality product robustness and the customer satisfaction. Now, and you see that this is endorsed by the US FDA, EMA, and other global regulatory agencies. And I think, and we all think that as the time is passing, many regulatory authorities throughout the world are going to adopt this particular concept, because it is the today's requirement. And maybe the quality by design approach and this concept is increasing day by day, because we are not dealing with the pharmaceutical product. We are dealing with the shortages of the products. We are dealing with the counterfeit products. We are dealing with the products that are not of the quality as we require. So we need a framework to design our products to design our product according to the present day requirement. Now, some of the advantages of the quality by design approach is that it minimizes the deviation. It would give us a high quality product, lowers the batch failures, it reduces the defects recalls. Sometimes we see the recalls, rejects, and improve the regulatory compliance. Now, if we talk about some of the demerits of the quality by design, the main thing is that we actually now do not properly understand the concept of the quality by design, and it is necessary for the regulatory authorities to simplify the concept of the quality by design, to simplify the terminologies of the quality by design, to give us to give us a guideline, to give us a simple framework, so that so that we train ourselves and we train our future generation of pharmacists to adopt it. So this may be some of the demerit, but if we compare the demerits with the merits, we find that the demerits outweighs the merits in and but at least we request that we need the guidelines as simple as possible, as simple as more flexible, so that it is adopted throughout the world in all the regions as well. Now, if you talk about the application of the quality by design, to keep in your mind that this is not only limited to the development of the pharmaceutical product. It is now also used in the analytical method. And I was reading a book which is related to the analytical quality by design. By it is applied on the drug substances formulation, dissolution testing by clinical trials and manufacturing, and many more which is coming as the time is passing. So the application of the quality by design is increasing day by day, and we hope many others to come in the later. Now, what are the elements of the quality-based design? So this is a figure which is adopted from a book, a very nice book which I read for the preparation of this particular lecture. I acknowledge the editors, the respected editors, and the topic is Introduction to Quality by Design from Theory to Practice. So you see that from this should give us elements of the quality by design. It shows that there are six components of QbD, starting from QTPP, which is quality target product profile. The second one, which is as important as the first one, is the critical quality attributes. Then we talk about quality risk management, design space, concept of the design space, then how we control ourselves in terms of the control strategy, and finally the continual improvement. So the clock and till continual improvement, but the process going on as we are going to target new drugs and the new drug molecules. We have to follow this in a scheme. Now, what are the elements of the quality by design? As I told you, the first important target, and this is the first element of the quality by design, is the quality target product profile. And when we are designing the quality target product profile or target product profile, sometimes we deal with the product, sometimes we are formulating a drug for the first time, and for that we have the target product profile. And later, as we get the data, as we get the studies from the clinical trials, we will move towards the quality target product profile. And at other times we deal with the quality target product profile in terms of the reference product. Sometimes the reference products are available in the market, and for the generic development we have to develop a quality target product profile by studying the reference product. So the quality target product profile, it is a summary. It is the summary of the quality characteristics of the drug products, and the purpose is to ensure the quality, taking into account the safety and the efficacy of the drug product. And it includes the quality, safety, efficacy, strength, bioavailability, and stability. It should be noted that this quality target product profile is not limited to the dosage form. It also includes other information such as the clinical studies, dosage strength, pharmacokinetic stability, drug product quality attributes, container closure system, etc. So this quality target product profile is the first and the most important document, a guiding document, and on the basis of which the journey begins. Okay, so this is very important. That is a quality target product profile. We should have a proper understanding of the quality target product profile if we want to make a quality product which is free from the defects all the time. So it is divided into three, as I told you that now I will focus. Now, before starting each element, I will give you some concept of the quality target product profile in terms of the oral tablet. For example, if you are considering to develop oral tablet or formulate oral tablet, what may be the quality target product profile element for the oral tablets? In that case, we have to see the dosage form, that whether you are manufacturing the uncoated tablets or the coated tablets or the hard gelatin capsule or the sustained release dosage form like this. Then we have to decide the dosage design, immediate release or the sustained release route of administration, its dose, quality attribute, packaging, pharmacokinetics, storage distribution, patient acceptance. So these are some of the quality target product profile elements if we are dealing with the oral tablet, and similar is the case with the other dosage form. Now we will discuss something about the critical quality attributes, and these are the attributes that are applied to the formulation of the tablets. And I am giving this in order to give you understanding that what is actually critical quality attributes, and if you talk about the tablet, what may be the critical quality attributes of the tablets in terms of the drug product. Now you see, and we all being a pharmacist, we have studied these terms several times. The first important thing is the And this thing which is very important. Number one is the porosity. It is a critical quality attribute for the tablet, and we know that it influences the mechanical properties of the tablet. When we have the high porosity, it leads the faster drug release and the improved bioavailability. Another important critical quality attribute is the disintegration. Although the disintegration is not directly linked to the dissolution, but it gives us information about how the tablets quickly release the drug. So this is very important because this is the starting point for the dissolution as well. And the important thing is the dissolution, like the porosity and the disintegration time. The dissolution is another important critical quality attribute which always considered, and we know that it gives us some information in predicting the in vivo performance of the drug. Although it may not be the same at time, it may not be the same because the internal environment is so complex, but it gives us some guidance, and we have developed the test to predict the in vivo performance like F1 and F2 test, etc. And likewise, all these three, the content uniformity, that is the API per tablet, it again is essential for the accurate dosing and efficacy. So this is something related to the drug product, and here we have discussed some of the critical quality attributes of our product in terms of the porosity, disintegration, dissolution, and the content uniformity. Now coming towards the materials, if we talk about the materials and if we talk about the critical quality attributes of our materials, of the material in terms of the API, in terms of the excipient, we should know all these things. And some of them I have listed here in order to give you some understanding that what characteristic may be used if we are formulating the tablet in terms of the materials which we are using. One important thing, is it is the polymorphism. It is the ability of a substance to exist in more than one crystalline form, and this will affect the physicochemical properties, particularly the solubility of the drug. And it is necessary to know that whether the drug exhibit the polymorphism or not. And the important thing is the particle size, which is particle size is another critical quality attribute, and it influences the flowability and the packing of the granules. And again, if we have the variation in the particle size, this affect the distribution of the API in the tablet matrix and affect the dosage and the efficacy. Another important thing is the moisture content. We know that the moisture content, some moisture is essential for the better compaction property for making a superior tablet. And increase in the moisture content, but however, the increase in the moisture content creates a problem. It affects the mechanical properties of the tablet. It lowers the hardness of the tablet, and again, the moisture content also affects the disintegration and the dissolution. So now here till now we have read something about the drug, about the product which we are manufacturing, and about the material in terms of the polymorphism, particle size, and a long list is there. So we have to study each and everything if we want to have the proper understanding of the material which we are going to use in our formulation. Now coming towards the next thing which we have to consider is the process parameters. What are the critical process parameters? For example, if we talk about the tablet, the first important thing is the size reduction, which we technically called as a combination. It is a process of particle size. It is the process of reducing the particle size of a solid substance into a final state of subdivision. That is the process of the micronization. It helps us to get a smaller tablets, easy to swallow, and ultimately improve the bioavailability and the compaction properties. And we know that most of the drugs available in the market and most of the drugs in the development stage have the solubility issues more than maybe the 40 or 50% drugs, maybe more, have the solubility issues. This is a very, very important topic and very, very important thing to consider when we are developing a formulation. Particularly, it has a problem. It has the solubility issues. The other important thing is the granulation. Again, it has an impact on the granules and the tablet quality. In this case, the critical quality attributes in terms of the granulation are the binder concentration, binder addition, how you are adding the binder, and what is the speed of the granulation. For example, when you are making a wet mass, dissolution, and this affect the dissolution and the pharmacokinetics. And the important thing is the drying. And again, when we are drying, we have to consider the temperature. So you see that if you are moving, if you are studying one thing, this is linked to so many things, because as I told you, that if you are talking about the granulation, so granulation is not a simple thing, because now here you have to consider the binder, you have to consider the binder concentration, you have to consider the addition of the binder and the volume, all are influencing your product quality. And similarly, is the case with the drying. You see, in case of the drying, you have to consider the temperature. What temperature, what may be the residence time, whether you are using the static bed dryers, or whether you are using the fluidized bed dryers, or whether you are using some other techniques, all are very important in terms of the temperature, in terms of the residence time, in terms of the end moisture which your product contain. This should carefully be considered, and this should be properly monitored. As finally is the compression and the compaction, the speed of the rotor machine, the compression force, all are influencing them. So you see that the making of manufacturing of tablet, which we are commonly consuming daily, you see the concept, and you see the complexity of the process. And if you move from one dosage form to another dosage form, so many things are there. We need understanding of all this if we want to make a quality product. So till now we have covered the three main things, that is the drug product, and then we have discussed something about the critical quality attributes of the materials. Then we have studied something about the process parameters, and I already told you that this is something which we want to learn. Although there is a long list, and we should have the knowledge of all these things if if we want to make a quality drug product. Now coming towards the risk assessment, this is also a new term. And risk, if we can divide this into three: what is risk, how you manage the risk, and how you assess the risk. This is the three different definition. It is a combination of the probability of the occurrence of the harm and the severity. Occurrence and the severity. So it is the risk, and how you manage: by analyzing, by prioritizing, and by managing the risk. And the risk assessment, and for this so many parameters are available, and so many things are available for the risk assessment. For example, this include for example, the basic risk management. It include the flow charts. Another is the FMEA, which I want to discuss something today: failure mode effect analysis. Another one is the failure mode effect and criticality analysis, FMECA, failure mode effects and criticality analysis, fault tree analysis, and so many others are there. These are the best tools of this assessment, but generally we are focusing more on the FMEA, that is the failure mode and effect analysis. Now, further simplify, and this is the thing which we also get from the regulatory authority, is the Ishikawa diagram. What is the Ishikawa diagram, and how we can use this particular? And then after this, we will move over towards the next, which is the failure mode and effect analysis. Now, what is the Ishikawa diagram? It is also known as the cause and effect diagram, or the fishbone diagram, because it resembles certainly to the fish skeleton. A team of the experts, they work together to develop a Ishikawa diagram, and the purpose of this diagram is to identify the potential variables that have an effect on the product quality. This can be drawn simply by making a box at the end and a horizontal arrow that shows the fish spine. Then the major categories and the problems are considered. There are further sub branches. So if you consider a diagram, this is the Ishikawa diagram, which is adapted from the Q8R2 document. And here the end product is the tablet, which is further divided into, you see, six major heading, that is the drying, analytical, raw materials, granulation, compression, and the plant factors. And each one is further subdivided For example, in case of the drying, we are considering the temperature, we are considering the relative humidity, airflow, and the shock cycle. Similarly, the analytical sampling and the method. So you can make an Ishikawa diagram of any problem you are facing. For example, in terms of the capping, if the capping is a problem, we place the capping at one end. This is our major problem, and then we make the branches and the sub branches, and then brainstorm the session to get the major cause of the capping or the sticking or the mottling, or any other disintegration or the dissolution. So many things are there. So the problem which we are facing, or the problem that are related to the formulation of the tablets, it can be brainstormed by using the diagram like this, which gives us the major causes. So you see that from this we got so many things, and from this help us in prioritizing that which one may be the possible cause of a problem. So once we sort out the problem, we can work on it. So once we get the information from the Ishikawa diagram, then the next stage we move. This is the risk assessment stage. And if I talk about the risk assessment, this is done again by a group of the experts, and they they rank the probabilities, detectability, as I told you, that they rank the probability and the severity and the detectability. And in that case, so you see that the first stage is to draw a fishbone diagram. The second stage is to use the failure mode and effect analysis. And in this analysis, we get a number which is known as the RPN number. That's the risk priority number. This is a numerical value, and this is based on the severity, occurrence, and the detectability. And in this way, you see, on the basis of this number, you see that from this you realize the thing, that the finished drug product, co-shifting, high shear wet conditions, sieving, lubrication. So on the basis of the Ishikawa diagram, we can prioritize, and on the basis of the RPN number, we can further divide it in terms of the low level of severity, medium level of severity, and the high level of the severity. So you see that in that case, we have identified that the granulation is the major problem. For example, lubrication is the major problem, and similar compaction is the major problem. So once we get these problem, which is highlighted from the FEA mode analysis, we are able to move on, and then we can apply this by using the design of experiment, to see the actual impact of this in our on our particular formulation. Now, coming towards the next heading, we are moving very quickly towards the end of the topic, and I hope that you are understanding this very well. Uh, and you will comprehend some information, and later when you read the Q8R2 document and the other literature available, you will be in a position to understand the concepts, and you will be able to apply it. Now, what is the design space? The design space, it is the range of the formulation and the process variables that are determined to be safe and effective. So, if you are producing a product, you have to decide, for example, the concentration level. For example, if I am using a polymer, if I'm using a disintegrant, so what may be the lower concentration of the disintegrant, and what may be the higher concentration of a disintegrant. So we in the design space, we know the range of the formulation and the process variables. So what is its effect on a particular outcome, and then this is then defined through a series of the experiment. And you see the design space. With the help of this picture, you can get the thing about the design space. This is the design space. You see the red region is the design space, which is covered by the control space, and finally the knowledge space, which we gathered. And on the basis of this, we have make the control space and the design space. So you see that how we are moving. and what is the actual concept of the design space. So we are controlling our design space by using a control space. Control space means the control strategy, how we are controlling the things so that we become in the design space. And you know that the design space, yeah, working with the design space is considered, it's not considered as a change. But if we move beyond the design space, it means that we need the approval of the regulatory authority. So this is the concept of the design space, which is covered by the control space and the knowledge space, which we get when we are designing our product. Now, how to develop a design space? So I told you that we have to start it from the quality target product profile. We have to start it with the quality target product profile. This helps us in establishing the design space. So first we study the quality target product profile, then we determine the critical quality attribute as we did. Then we have to perform the risk assessment as we did in case of the Ishikawa diagram and the FME mode analysis. And then finally we have to conduct the design of experiment, evaluate the experimental data, and we may conduct the additional experiment, etc. Now, in order to further understand the concept of the design space, I'm showing this, and this I got from the Q8 or 2 document. This is related to the design space and the various type of the plots which we are using for the proper and the clear understanding of the process. So you see that this is this is, you see, this is the response surface plot, and the response surface plot. So you give the design space in term of the response surface plot, in term of the contour plot. So response surface plot, we have two parameters. So these parameters are parameter one and parameter two, and we are going to see the effect of this parameter on the dissolution. So if the dissolution we want to be 80%, what may be the level of the parameter 1 and parameter 2, parameter 1 and parameter 2 to get the 80% dissolution. So you see that initially when they are when parameter 2 is at 0 and the parameter 2 is at the high level 60, we got a lower dissolution rate, which is near to the 60%. And again, if we see that if we are increasing the parameter 1 at the 40, and for example, if it is at 40, we are getting the 75% dissolution. And similarly, if we see that in case of the parameter 2, if we are increasing its concentration, again we are getting the lower dissolution rate. So this graph gives us a clear understanding of the process of the two variable in terms of the z region. So this is the this is, you see, is a 3D plot like this, in which we are taking 2 on the x and y response on the y-axis, and checking the behavior of this parameter on a particular outcome. Now this is the contour plot. This is also adopted from the Q8 or 2 document, and this also gives us the space which we want to have for the parameters in terms of the parameter 1, in terms of the parameter 2, and the shaded line clearly indicate then when the region is in the dark, we are getting the resolution in terms of 60%, 60 to 65%, 60-65%, again 70 like this. And when we are going from the darker region to the lighter region, we are getting the higher dissolution rate, which ranges from the 85 to 90%. So from this contour plot, we got an information that the lighter region, it gives us the information that sorry, that this may be our design space in which we feel ourselves comfortable because we are meeting our target, that is 85%. And you see in the last, again, this is the design space for the granulation parameters, and it gives us the information that what may be the suitable concentration of the parameter 1, what may be the suitable concentration of the parameter 2 so that we get a dissolution which is equivalent to 80% or more. So you see that the design space help us to reach our target in formulating a product whose resolution is 80%. Now, as I told you that once we get the concentration, because at the start we set the we don't know what concentration should be used. So by brainstorming, we got the parameters that are creating the problem, and then we have to put the concentration of our independent variables in the design of experiment, which is also a systematic tool, and it helps us to identify the critical process parameters. And we all know that the design of experiment, various type of the design of experiments are available, that include the factorial design, partial factorial design, central composite design, and so on. So according to our requirement, we select a particular design, and in that case, you see that what may be the in that case, in the design of experiment, we have to define the problem. What is our problem? What are the responses? What are the responses and their measurement? We determine the factor and the level, which is very very important. Which factors we are using and at which level? Because if the levels are, if the factors level are closer to each other, then we may not get a significant effect. So it may be somewhat far away, so that we get the thing that maybe in the maybe it's significant. And then we have to select a suitable design, conduct the experiment, get the data. The data is then put up in the software, and then it is analyzed by using the various plots like the Pareto charts, like the half normal plot, like the interaction plots. In this way, once we get the information, then we apply the ANOVA, and which give us information that whether the model is significant or not, whether we have to use the linear model, whether we have to use the quadratic model. Then we interpret the results, and on the basis of this, we reach us to our design space, which is our ultimate objective, to make a quality product. So another important thing is that when when we have developed a product, and now we know many things about, this is not sufficient. We have to control formulation process throughout the manufacturing, and throughout the manufacturing as well as after manufacturing. So what will be the control strategy? The control strategy, we got a formation from the regulatory authorities. That by the control strategy, it means the plan set of the control drive from the process and the product understanding. So we have to check the input. So if we want to control our product, we have to check the input materials, for example the APIs and the excipients, the packaging materials, the finished product testing, the finished product standard procedures, facility control is also important, because simply controlling the materials and things and the packaging is not sufficient. We have to control our facility in terms of the environmental setup, in terms of the temperature and the relative humidity, in terms of the unit operations which we are moving, how efficient these unit operations are, and for example the particle size distribution, drying, etc. So for this, it is necessary to have the standard operating procedures, and the guidelines. So and each and everyone should follow those guidelines in order to optimize the control strategy in developing a quality product. And finally, the continual improvement. This is by this we mean that the quality of the product can be enhanced throughout the life cycle of the pharmaceutical product, as we get the knowledge and as we get the experience, as we are manufacturing the product. We have to continually improve the product, so that it is within the design space, and continually improve our product as the time demand. So this is all about today's short, small session of quality by design. And this is finally our conclusion. This will be my conclusion. Quality by design is a systematic approach for the development of a pharmaceutical product. And the key elements of the QbD are the QTPP, quality target product profile, quality risk assessment, design space, control strategy, and continual improvement. And if we follow this systematic approach, the formulation scientists develop a quality product that meet both the manufacturer's requirement, the patient requirement, and I must say the regulatory authority requirement as well. But at the same time, there are something which we want to emphasize. As I want to emphasize, is the important is simple regulatory guidelines. We at the time today we need simple regulatory guidelines in order to apply it in order to give its importance to others, to implement it in various research domains. And finally, our main objective is to train our future pharmacists, the future generation of pharmacists, more knowledgeable and more acquaint themselves to the latest requirement. Beside this, I also must say that artificial intelligence also have a role in various domains, and maybe next time, maybe the regulatory authority also consider the inclusion of the quality by design for the formulation, product development, and optimization. Thank you so much for your listening and for your attending this webinar. I tried my best to give you the key concept of the quality by design as simple and possible. And as you move and read the guidance for the industry Q8 R2 document, which is available. Similarly, as I already told you, that I read a latest book available from the Springer Introduction to Quality by Design. Many points and many figures are taken from this particular book, a very useful and simple book. And I appreciate the efforts of the editors and the authors who have contributed in the publication of this particular book. Thank you so much.
Thanks for joining this webinar. Those who have already registered, they are all requested to fill the post-webinar evaluation form, which will be given in the description box and will also be shared to you in the webinar WhatsApp group and our official WhatsApp channel. Follow and subscribe to our YouTube channel. You can also join our official webinar group, which is given in the description box. Those who registered already will get the certificates soon. You will be informed in the official webinar group and also in our WhatsApp channel. All links will be given in the description box. Join us for there. Stay happy, have a great day, and I'm so thankful to Pharmacozyme for having me here. Stay tuned to Pharmacozyme for more webinars. Allah Hafiz, and thank you so much.